we're talking about endospores, and one of the things that we find in endospores, one of the chemicals unique to endospores is dipicolinic acid. The dipicolinic acid structure is shown here. Okay, and you see it's got the carboxyl group, so it's got the negative charge, so you can put two of these together and they can interact with the calcium. They have done a lot of research on this, and they believe that the calcium dipicolinic acid complex causes dehydration. And one of the things that happens that makes that endospore very resistant is it loses water. Its water content goes down. Normally water content's around 70%, goes down as low as 60%. So that dehydration makes the enzymes less active, makes that form, that endospore, much more stable. The other thing they believe it does is it intercalates between the DNA bases. That means it kind of, intercalate means it gets between the bases and stabilizes DNA to prevent denaturization. So basically two functions: causes dehydration, increasing the stability then of the DNA that's being stored within that endospore. So we look at the overall cycle. We have a vegetative cell. It's a normal cell in a normal environment where it has all its carbon and nitrogen, everything it needs in the right environmental conditions. That cell will give rise to two cells. It'll grow, and each of those two cells will grow larger and divide into 4 into 8, 8 to 16 to 32 to 64. They're going to be doubling every generation. And if, for example, Escherichia coli has a generation time of 20 minutes, every 20 minutes it will grow and reproduce. It will double. So 1 goes to 2, 2 to 4, 4 to 8, and so on. That's called growth reproduction, normal growth of a microbe. If something occurs in the environment that makes that environment disadvantageous to certain microorganisms, certain genera have the ability to respond by synthesizing within this bacterial vegetative cell a highly resistant structure known as an endospore. Knowledge of the existence of endospores is very critical in terms of the food industry and making sure food is safe, because you have to make sure some of these bacteria like Clostridium botulinum that can be the endospores can survive a heat treatment are not present in the food. So you have to really ensure that. So knowledge was very important, and these structures are highly, highly resistant. So that microbe then, instead of growing and reproducing, it commits itself to the synthesis of an endospore within the vegetative cell. When this endospore is fully mature, eventually that bacterial cell lyses, releasing the endospore into the environment. At this point, I like to call it a free endospore, just to distinguish the endospores of bacteria from the spores of fungi. The spores of fungi have a major goal: reproduction. You know, you learn, if you had general biology, you may have learned about the sexual and asexual spores of fungi and how they're involved in reproduction of fungi. Well, in bacteria, the endospore is not a reproduction mechanism, it's a mechanism for survival. Because in the spore cycle, we will start with one cell, that cell will synthesize an endospore, it will lyse, releasing the endospore, and that endospore will, what we call, germinate and give rise to a vegetative cell. So we start with one, we destroy it, we make one, so we end up with no increase in the number of cells. We start with one, we end up with one. So it's not for reproduction. Now, this process here, where this endospore, when environmental conditions change again and become beneficial for it to undergo vegetative growth and reproduction, this endospore will go through a process, and I want to call this whole process actually germination. Your book divides it into activation, germination, and outgrowth. And in reality, you know, where do you stop one and start the other? I think it's better just to call the whole process the endospore germinating and giving rise to a vegetative cell. So you'll see initially a small outgrowth occurring. That outgrowth will get bigger, okay? And eventually it will produce another vegetative cell. What that tells you, if you think a little bit, is that within that bacterial endospore, there has to exist the full complement of DNA, all the genes. The double-stranded DNA molecule has to be trapped in there, right? Because for that endospore to give rise to a vegetative cell, it's got to code for all the enzymes that are used to make all the macromolecules to make that cell again. Okay? So we will definitely see DNA in the endospore. Okay, this is some figures that are supposed to kind of show you the different steps. I'm just showing you just to show you how things change with time. If you start out with a free spore, and I like to call it once again a free endospore, okay? I would call it free endospore. Okay? It's going to be highly refractile. That's phase contrast, okay? And you see how highly refractile it is in comparison to this one. Okay? Basically, part B is supposed to show you that activation has occurred. So when you first get a spore to commit toward germination, you will see its physical properties change. And you see them very clearly here, and that's why I'm showing them to you. And this is showing you how the vegetative cell is outgrowing from the endospore, and you see it's outgrowing even further. It's pretty close here, almost splitting this off and becoming a cell. So in your book they call C and D outgrowth, they call B activation, and there's one step in between that they call germination. Do not worry about that. Okay? We're going to call the process germination. We're going to continue to call it germination. So germination is the process where you go from a mature endospore that's out in the environment, and it gives rise through the germination process to a vegetative cell. Okay, this is the spore cycle, and this is the one that's in the new edition. The old editions of the book, the 12th edition, has a lot more details, and I prefer this one because I think it shows you the steps a little more clearly. But we'll go, the next set of slides will be the one from the 12th version, and I'll go through that just to point out some of the things we're going to see here. So if we look at this being a vegetative cell, normally that cell, if it's in a nice environment and it has everything it needs to grow and reproduce, that cell will increase in size and divide into two cells. Okay? That's growth reproduction. Those two will divide and produce 4, 4 into 8, and pretty soon we'll have millions of cells in the population. Okay? But if something happens in the environment, on that spore senses, they have ways in which the bacterial cells can sense changes in the environment. It will commit itself to the synthesis of this highly resistant structure called an endospore. And what you'll end up doing is, one of the things that will start is you'll get an invagination of the cell membrane that will cause you to have a pre-spore here, and this separation between them is called the septum. And so what you're going to see is that septum is going to grow around that pre-spore, or you can call it a forespore, to eventually form the basic endospore. So it's sort of like this thing will go around. It's kind of like engulfment. You know, it's going to go around and spread around and engulf that pre-spore, forming the endospore. And of course, then you've got to go through all kinds of steps to add more and more layers. You get cortex formation at one stage. You then end up adding the sporoscote at another stage. You see the concentration of calcium dipicolinic acid increase as this thing matures. So you see dipicolinic acid. You see what we call small acid-soluble proteins that grow up in high number. And this spore will continue to mature. And once it's mature, this is showing the final maturation. This would be one that's at the end of this cell. That'd be a terminal spore. And once it's matured, it will retain the malachite green. Remember, to stain the endospore, you have to force the dye in by steam because it's dye resistant. Once you get it into the endospore, whether it's a mature endospore within a sporangium or whether it's a free endospore, once it's mature, it will retain that malachite green. If you try to wash it off, it'll wash out of the vegetative cell. It will wash out of the sporangium, which is the vegetative cell surrounding the endospore, and they will stain with a counterstain pink. So if you have an endospore that's mature within the vegetative cell, basically within the sporangium will stain pink, the endospore will stain green. Once the endospore that's mature is released, the bacterial cell lyses, the endospore also stains green. Regular vegetative cells, because you steam it to get it in, and you don't even have to do that to get it in the regular vegetative cell, but it washes right back out. So vegetative cells also stain pink. If you're doing a spore stain, this would stain pink, these vegetative cells would stain pink. Once this stain matures, and it probably has to go up in somewhere in these stages here, then it will retain the malachite green and stain green. But the sporangium will always stain pink. Okay, these are the more detailed steps in endospore formation, and you're not going to be responsible for these, but I thought it showed you a little better that you get invagination of the cell membrane to form this septum here, okay? And then you get, in other words, the cytoplasmic membrane invaginates. Then you have this cytoplasmic membrane kind of grow around that forespore. Okay? So you see it growing around, and eventually it becomes an actual endospore within that vegetative cell. Okay? And then you add all the layers. You add the exosporium first. The primetal cortex is added, and so on. So all the various layers we talked about before are added, and the thing continues maturation, and once it's finally mature, that cell will lyse and release the free endospore into the environment. It's just another look at it. So the only one you're required for is the first one that I showed you that's in the new edition. Okay, small acid-soluble proteins. These small acid-soluble proteins, remember, they appear in high concentration at the same time as dipicolinic acid is increasing in concentration, complexing with calcium, making that cell more dehydrated, more mature, because as it's more mature, it becomes more and more dehydrated. So there are basically a couple of functions with these small acid-soluble proteins. One is they can later on serve as a carbon and energy source for the outgrowth of a new vegetative cell. You're not going to make macromolecules without inputting energy, okay? So you have to have a carbon or an energy source. You need to make carbon to make the organic molecules. Typically that organic compound, these little proteins, also are used as the energy source. So these provide the carbon and the energy to make a new vegetative cell. So that's their function there. So it basically provides that carbon and energy for the germination process. And once again, I said the book divides it into activation, germination, outgrowth. We're going to call the whole process germination. Okay. The other thing it does is it makes DNA more stable. It binds to DNA in the core. Remember, the DNA has to be in the endospore. That endospore didn't contain the DNA. You couldn't give rise to a vegetative cell. You don't have the genes to code for all those proteins that are necessary for that process. So it protects DNA from damage by UV radiation and also from drying. It changes the molecular structure of DNA from the normal DNA, which is the B form, to an A form. A form is more compact. You'll learn in molecular biology 1, those of you who are biomedical majors, that you have an A form of helical form of DNA, you have a B helical form. The B helical form is the normal form. Also, RNA is more like an A. Okay? But it changes it to the A form. A form is more compact and is much more a resistant form where the DNA is much more stable. The A form is also more resistant due to the formation of, it's more resistant because it prevents the formation of the pyrimidine dimers. If you have DNA and you have two thymines that are next to one another, what kind of base is thymine? It's a pyrimidine, okay? It's only got one ring. If you have two thymines next to one another, they can form a covalent bond between them. That's one of the things UV light does. In fact, it does it in our own DNA. That's why I have so many skin cancers. It's been first semester that I've gone this far without having my nose either bandaged from a skin cancer or coming in all bandaged up on my arm or having my foot or whatever, because I've had so many. Native Floridian, and we've got the beach, Grandpa's beach house, 2 months in the summer. I'm paying for it now, gang. So warning you, wear your sunscreen. Even though some sunscreens have been shown to be a little carcinogenic themselves, but I can't wait for losing, you know. No, they really did. A lot of the early ones, they were carcinogenic. You know, they've shown in studies that they can promote cancer. Okay, even the best one now, the zinc ones, they're talking about light interacting with zinc, forming some kind of zinc-free radicals. So I'm saying, oh gosh, we can't win. But anyway, stay out of the sun. Put the sunscreen on, because I think the UV is more carcinogenic than the carcinogens in the sunscreen, so I still would use it. But anyway, the way UV causes damage, if you have two thymines adjacent to one another, ultraviolet light causes them to be covalently linked, causes what we call a thymine dimer. Thymine is a pyrimidine, and so that's why they say it causes the formation of pyrimidine dimers. Okay, and there are other pyrimidines can dimerize, cytosine and so on. So it basically, it's a mechanism of mutation. It can cause malignancies with time, especially if you get multiple ones. But and since it causes mutations, then that would make the DNA of the endospore not be too healthy. Okay, so this form, the A form, is more resistant to formation of these dimers. You don't get them as frequently, so it makes the DNA a little more stable. Okay, this just shows you some locations of endospores. We used to use that information sometime to help us identify bacteria. Typically, we use all those biochemical tests that you're doing now to help us identify microbes, staining procedures, biochemical tests, and so on. But I just want to show you they can be located different places. Here's one that's clearly central. You know, it depends on how big they are when they're dividing it. Sometimes they'll appear a little bit to the side, but typically these are more centrally located, so they call them central spores. If they're near one end, away from the center, near one end, but not at the very end, they sometimes call them subterminal spores. And if you have them right at the very end, they almost look like little tennis rackets, you know, a little racket at the end, then they're called terminal spores. But once again, we don't use that, and I'm not going to ask you to know these names. Okay, this is a nice summary diagram, and it shows you some of the different properties that we can actually see these different forms. This would be a vegetative cell, undergoes stress, it starts developing an endospore, and as that endospore matures, okay, then it becomes dye-resistant, heat-resistant, and so on. And as it finally matures, this bacterial cell lyses, releasing the mature endospore into the environment. And if we look at this figure, we can see here's a good example of a mature endospore here. I think there's one there, okay? Now we can look here, you can see it's within the sporangium. It's within the sporangium. These are endospores that are fairly mature, that are within the vegetative cell. And all these other cells that you see, no endospores, those are just the normal vegetative cells. So not only do we see them in a spore stain as being different, but we can see them when we do phase contrast microscopy as being different. You can actually see the difference between endospore and the endospore within the sporangium. Okay, let's look at microbial locomotion, meaning movement of bacteria. One of the major ways bacteria move is by use of a flagellum, but we'll talk about a couple of other types of motility, gliding motility, and I'll mention one to you. It's not even in the PowerPoints, just so you know have the knowledge that there are even others that the book doesn't even necessarily mention. And that's called contraction of an axial filament, but we'll cover that later. Microbial locomotion with a flagellum is a little bit different between eukaryotes and prokaryotes, and we'll get into the differences in flagellum structure between a prokaryote and eukaryote, and the differences in the way these flagella promote movement. But before we do that, let's look at how flagella can be arranged around a microbial cell. The arrangements can be polar, where the flagella are at one end of the cell, okay? Some cases it'll be both ends, okay? You might have another tuft here. So if it has a single flagellum at one end, anytime it's at one end, we call it polar. Okay? I should say at one end or both ends, because there are some cases where, but it's at the poles. It's not all around the entire periphery. So polar is in contrast to this one, where you see it's all the flagella are scattered around the entire surface of the cell. So if it's at one end and only one single one, we call it monotrichous. So there's your term, monotrichous. Mono means one, trichus is here, one here, so one little flagellum. Then we have a little bundle or a tuft of them, okay? Loco means a tuft, okay? So it's a bundle at one end, okay? We can also have them, in fact, once again, trichus means here. We can also have them just like this, except another bundle at the other end. And when that happens, you call it amphitrichous, meaning a tuft or bundle at both ends. And then the other arrangement is to have it scattered around the entire periphery of the cell, and that's called peritrichous. So I think of peri being periphery, but actually peri means around, okay? So it's around the entire cell there. So that's peritrichous. Escherichia coli that you deal with in the laboratory, Enterobacter aerogenes, they move by flagella, and they have peritrichous type flagellation. There's another one you deal with, I'm not going to ask you that. There's another one we deal in the lab, I think it's Pseudomonas aeruginosa. Pseudomonas has a polar flagellum. Okay, this has, I added these notes here. Most of what you see here are going to be on the next page, but I wanted it side by side. And wanted to show you basically three parts to the flagellum. There's the filament, which is what's actually moving, okay, what's actually rotating and moving. There's the hook that connects that filament to what we call the basal body. So if you look over here, everything from here down to here, this whole region here is basically the basal body. The basal body consists of 1, 2, 3, 4 rings. Okay. Now, is it 4 rings in all microbes? What is this a gram positive or gram negative? Can you tell? Right. Why is it a gram negative? They have the LPS layer. Okay. It's a true paraplasm. So this L ring that we see here is in the LPS layer, so we don't see it with gram positive. So you have at least 3 rings. In gram negatives, we have the 4 rings. Those 4 rings and the rod going through them are called the basal body, and that's the rotor. Bacterial flagella move like a propeller. When you learned about eukaryotic flagella in maybe general biology, remember it has that wave-like motion? okay? This one's a little prop, it's just moving just like the prop on your boat, okay? So it's much different in terms of the way it moves, it rotates, and that's why you see on this diagram, see how it's showing it rotating?